So our next speaker is Dr. Wayne Chadwick. He's coming to us from Healx, and he's going to talk to us about the power of AI in drug discovery and how they found some repurposed candidates in order to think about how we can treat Angelman syndrome. So here we go.
Perfect, thank you. Right, hi everyone. My disclaimer is I work for Healx, and at Healx we do drug discovery a little bit differently. We use artificial intelligence and machine learning technology to predict which drugs could potentially treat a rare disease. And these are drugs which are already on the market. They're approved for different indications, and we use our algorithms to determine which of these drugs we could reposition into a particular rare disease, such as Angelman syndrome. We've got about a 50% hit rate with the drugs we predict for each of our particular rare diseases we work on, and we uncover typically between 1 to 5 novel mechanisms for each of the diseases, including Angelman syndrome. We are a clinical stage biotech company. We focus on rare and pediatric oncology as well as rare neurology. And last time I spoke to you, I gave you an update on our lead candidate for Angelman syndrome, which is 0553. This is an existing drug again, which was approved for a separate indication, which we were hoping to reposition into Angelman syndrome. It was really potent. It was really impressive. It improved multiple symptoms or phenotypes in the Angelman mouse model. The only problem was it was really designed for short-term use and wasn't really designed for long-term chronic use. But we were confident that we could overcome that problem by reformulating the drug, and that means changing how the drug is absorbed by the body and distributed throughout the body. And we did this with our partners, TRX Bioscience, who had a lot of experience in reformulation. However, it became quite evident early on that the physical chemical properties, basically the structure of 0553 was somewhat unique, and it didn't allow it to be reformulated correctly. Basically, it wasn't stable when we dosed it in the animal. And because of this, we sort of had to abandon it, which was unfortunate. But we were really confident in the mechanism which was driving or promoting or improving the symptoms in the Angelman mouse. So we thought we'll go back and look at other drugs which have the same mechanism to see if we could identify a drug which was as effective as 0553, but maybe was able to be dosed over a long period of time, or would allow us to reformulate it so that we could dose it for a long period of time. But we needed a way to compare these drugs with one another, and for that, we needed to set up an in vitro screen, which we've heard about today. So for this, we used Angelman mouse neurons which have a unique morphology. They have very different type or different structure, particularly with their dendritic spines. Now, dendritic spines are basically just the tiny little bumps on the branches of neurons, and they're very important for neurons to communicate with one another. Now, these Angelman mouse neurons, like the human neurons, have fewer dendritic spines, and the dendritic spines that they do have are mostly immature, and there's very few mature dendritic spines. And these factors means that the Angelman neurons are just unable to communicate as effectively as a healthy neuron would be. And we knew that 0553, from the previous work we had done, was able to not only increase the dendritic spine numbers, but also promote the maturation of these dendritic spines, so that these neurons, after treatment, were indistinguishable from healthy neurons in terms of their morphology. So we identified 18 drugs which had the same or similar mechanism to 0553, and we tested them in this assay. And we identified 10 drugs which were highly potent or as potent as 0553. And then we further sort of triaged these drugs. We looked at their chronic use potential, their distribution profile, if they got into the brain at high enough concentrations, and their reformulation and commercial potential. And we identified from this, we landed on 3 drugs which we wanted to investigate further. One was an active metabolite of a drug which is currently on the market. The other one was a shelved asset, and that means that this particular drug had actually reached a phase 3 clinical trial for a separate indication, but the company which was progressing it decided to abandon it for commercial reasons. But it was safe, and it was actually useful, and it can be used chronically, so it doesn't need to be reformulated. So that's quite exciting for us, because that means this drug can transition into Angelman syndrome quite quickly. The other is another drug which is on the market already, but it's only available through prescription. So this is the data of the dendritic spines. I won't go into it, but suffice to say that all 3 drugs improved the number of dendritic spines in the Angelman neurons and also promoted maturation of these dendritic spines, so that these neurons were basically indistinguishable in terms of their morphology from healthy neurons. And that means that these neurons could effectively communicate better. But next we wanted to see how these drugs actually fared when we dosed it in an Angelman mouse. So we did this. We dosed the drugs for around 2 weeks prior to putting them through a battery of behavior tests. We looked at motor deficits, so we put them in an open field and assessed their mobility. We assessed how many marbles they could bury. We also looked at their grip strength, fallen grip strength. We assessed motor control, and we looked at their balance and coordination on a rotor rod, so seeing how long an animal can stay on an accelerating rod. And this apparatus also allows us to measure motor learning. And then we also measured their cognition by using the object recognition test, so seeing how the animals are able to differentiate between 2 objects. So this is the data, so let me just orientate you to these graphs. The white bar are the healthy mice, and that's what we're trying to achieve with the drug treatment. The gray bar is the Angelman untreated animals, and we kind of want to move away from that level as much as possible with treatment. So if we just look at the distance moved in the open field, we see that Healx B and Healx C treatment drugs increase the mobility of the animals in almost comparable to what we see in the healthy animals. Surprisingly, Healx A was not as effective. But all 3 drugs were able to increase the ability of the animals to bury marbles, significantly better than the Angelman untreated group. And again, Healx B and C performed much better than Healx A. Surprisingly, none of the drugs improved the grip strength, but all 3 drugs were able to improve the motor control in the animals by looking at the clasping. And again, Healx B and C were much more superior, improving the motor control to comparable levels that we see with the healthy animals. Now, this is really impressive to me. This is the rotor rod assay. And in this graph, the dotted line represents the Angelman untreated group, and then the gray bar on top, or the gray line on top, indicates the healthy animals. And if we just focus on day 1, we can see that all 3 drugs significantly improved the balance and coordination of the animals. So the animals were able to stay on this accelerating rotor rod for a significantly longer period of time. But what's really impressive is, again, Healx B and C groups, their balance and coordination is indistinguishable from the healthy animals. And then if we look at each day of testing, so if we test these animals on the rotor rod for days 2 to 5, we see that the balance and coordination of these animals improves with each day of testing for Healx B and C. And this is indicative of improved motor learning, which again is really, really impressive. Healx A, again, was less effective at this. And all 3 drugs were able to improve cognition. So all 3 drug treatments groups were able to distinguish between 2 objects. So we got an improvement in recognition memory here. So just to conclude, Healx B and Healx C, Healx B being the shelved asset, Healx C being the prescribed drug, were able to restore motor control, coordination, and memory in the Angelman mouse back to levels that we see with the healthy animals. And this is really impressive. We've got 2 drugs which could potentially move into the clinic quite quickly here. Healx A was less effective, and improved a certain amount of phenotypes. And just to reiterate, Healx B is suitable for chronic use. We don't have to mess around with reformulation for this. So again, it can move quite quickly into the clinic. Healx A and C does, however, require reformulation. But the good news is we've already started some of that reformulation work, and the reformulation is stable. And we've already seen improved amount of drug in the brains of mice with the reformulation. And we're hoping that that equates to improved efficacy, particularly for Healx A. So just to say again that Healx B and C look promising, and we're hoping to be somewhere close to being in the clinic by sometime next year. But it all depends on licensing discussions with the holders of the shelved asset. So thank you.
See, so I think if you didn't understand in vitro and in vivo, you would have no clue what he just said. But because you listened so carefully this morning, that was very clear that we have potentially some downstream targets that could affect, because this is not replacing UBE3A, but that could have an impact on some of the symptoms of Angelman syndrome. So that's exciting, very promising. And when it's a repurposed drug, it may have already been through some of the safety assessments that allow you to get from mouse to human a bit faster. So that's also very exciting.